So it's a pleasure to be here in Norwich and talking for the new Phytologist Next Generation Scientist Symposium. I'm definitely not one of these senior people that you've been hearing from. I'm just a new group, I'm very thankful for the opportunity to share our research with you today. So what my group is interested in is plant developmental plasticity. In particular, how plants can change their morphology to suit the environment in which they find themselves. So one example of this developmental plasticity is very early in the plant life cycle. So if a seed germinates under the soil and it's dark, it's going to have a very different morphology from a seedling growing in a full light environment. So this was nicely introduced by Lot yesterday, where we have this etiolated growth in the dark, with the seedling stem, the hypocotyl is elongating very rapidly to push that pseudobucca meristem in the shoot towards light so it can start to unfurl and start doing photosynthesis. So this is a nice, interesting example of different morphologies, but it actually gets more interesting if you start considering this at the cellular level. So if we imagine a cell in this hypocotyl, it's elongating very rapidly. Well, in order to know that it should elongate, it has to do a couple things. It has to integrate endogenous signals. So I'm a hypocotyl cell about here with environmental signals, in this case, the absence of light. And then it has to activate programming that would suit that situation. So what I'm describing here is a signal integration of endogenous environmental signals that has to occur. That information has to get processed, and then it has to be transduced into activation of coherent and appropriate downstream developmental and physiological programs. And so if we actually think about it, all the cells in the plant are doing this all the time. They're integrating all these informations, they're making decisions about what to do about it, and then they're activating programs. And so what we're interested in is how the phytohormones serve as oftentimes these signal integrators and program activators. So many things can influence the levels of phytohormone in a given cell at different stages of the biochemistry. And then, of course, phytohormones are master regulators or important regulators of many different processes. So in the example I just talked about, which is seedling growth and this elongation of that seedling stem, so one of the key hormones involved there is gibberellin, or GA. And so I'll focus the talk today on this particular hormone, GA. And you can see that GA is a major growth regulator in plants very easily by just simply spraying it on different parts of the plant. So this classic example where you can add GA to grapes and get bigger, juicier grapes that are, this has agronomic relevance in the field. In this cabbage, if you add a GA, you can get earlier flowering and these elongated stems. And this is a pretty dramatic example. But we can also see these effects in Arabidopsis. So if we grow Arabidopsis in a microtiter plate, you see normal Arabidopsis development. If you add some GA to that to a microtiter well growing Arabidopsis, you see a different morphology. You see longer petioles, larger cotyledons, larger leaves, grossly changed morphology. And that brings up a point I'd like to emphasize, that generally, even though this does sometimes work, broad manipulation of hormones is a blunt tool. Because hormones regulate so many downstream developmental and physiological processes, simply raising or lowering the levels of a hormone broadly in a plant is not a really good intervention strategy for helping our crops survive and thrive. And so what we'd really like to do is understand what's going on in the endogenous level. Where are the hormones? Where is gibberellin localized? And what is it doing in each individual case? To start to try and pull out some of this major reprogramming that the hormone can do on the global scale into very specific examples. And so in the case of the etiolated hypocotyl, we'd like to ask the question of where is GA distributed? So which cells have higher GA than others? And then how does that GA distribution relate to growth? So is higher GA always correlated with higher elongation, for example? And then finally, how is the GA itself regulated? So what are these signals integrating upstream of GA to generate the actual extant GA distribution in vivo? And so one of the ways we can start to access some of this information, in particular the GA distribution, is to ask where is the expression of the biosynthetic enzymes. And so GA3 oxidase is the last step of GA biosynthesis in Arabidopsis. There's 4 isozymes, and I'm just showing you the promoter Gus expression of 2 of them, GA3 oxidase 1 and 2. These are very important ones. And you can already see that there's some pretty interesting expression patterns. And contrasting, in fact. So the GA3 oxidase 1 is highly localized in the embryo at the what will be the shoot apical meristem, and then the root apical meristem for GA3 oxidase 2. And this continues throughout development, this contrast and this very specific expression patterns. In the anthers, you have GA3 oxidase 2, and then in the anther filaments, the GA3 oxidase 1. So there's clearly some control going on, and that might say, okay, this is where the GAs might localize, but really that's not actually going to tell us where the GA is localized. Because GA levels in a given cell or tissue are controlled by many biochemical and transport activities. So you have de novo biosynthesis within the cell, but you also have catabolic steps. Potentially, you can have reversible conjugation steps or compartmentation steps. And in particular, for the hormones, you have mobility, right? These are mobile small molecules. So export and import from other cells or tissues can also play a role. So it's really the ensemble of all these biochemical activities that's generating a distribution of GA in vivo. And so what we really like to be able to do is measure that distribution in situ, in a living plant. And so the way we go about doing this is to try and generate a GA biosensor. So when I was a postdoc in the Frommer lab back in California, I sought out to generate FRET biosensors for hormones. And so just a little background on how these FRET biosensors work. There's a ligand sensory domain that interacts with the ligand of interest, and that's fused to two fluorescent proteins of a FRET pair. Shown here are CFP and YFP. And so when the ligand sensory domain interacts with the ligand of interest, it will change its conformation. That will generate transduce into a relative change in distance or orientation of the two fluorescent proteins. And that's important because that distance and orientation change can change the amount of FRET between them. So FRET is just an energy transfer from a donor molecule, in this case CFP, to an acceptor molecule, or YFP. So shown here is after binding, you get a higher FRET state, and before binding, the lower FRET state. And the way we read this out is just to actually take the emissions of the two fluorophores after excitation of the donor. So if you just excite the donor, you get the cyan emission and the yellow emission. And you can see that after binding in this cartoon model, you get higher yellow emission compared to cyan emission. And so that ratio of the two can serve as a proxy for the presence of the ligand in the solution of interest. So how do we go about making a FRET sensor for GA? So the strategy starts with getting a sensory domain, so a ligand sensory domain that will interact with the ligand of interest, in this case GA. And so what better tool than to actually take what the plant is using to interact with GA, the GA receptor proteins from Arabidopsis. And so here's a structure of GID1 bound to GA4, and this is a Arabidopsis GID1 protein. And just part of the GAI protein, which is a delta protein, but we've removed off the C-terminus and just including the delta domain of this, the GAI protein. So this is a model of what we kind of started with as far as potential sensory domain, and it's a cooperative domain. So if the GID1 protein interacts with GA, it will fold, and then the delta protein could come along and sit on top of that. And so if you fuse that into one protein, we could get a clamshelling motion, and that could be the structural rearrangement that we would like to have for a FRET sensor. And so the second part is to put the fluorophores on, and so I'm showing here just variants of CFP and YFP. And so I did a screen where I combined a series of different FRET pair variants combinatorially with a series of different sensory domain arrangements. So whether the GID is first or second, whether we do truncations of this size or that size. And then, of course, we have different linkers to put the whole thing together. And this will make a single transgene, a single synthetic protein that hopefully will do the sensing for us. So I'm skipping over quite a bit of work, but then at the end, we ended up with a functional sensor we call gibberellin perception sensor 1. And this is very similar to what I just showed you, except that we have the linkers involved. And when we put this protein, purified protein in vitro, we can then ask the question, what happens before and after we add GA? And so in gray is before we add GA, and you can see if we excite the CFP, we take a fluorescence emission curve across various wavelengths, you can see there's quite a bit of emission already in the yellow, which means that some of the energy we're exciting CFP with is being transferred to YFP and being emitted as yellow. But when we add GA, we see an increase in the yellow emission and a concomitant decrease in the cyan emission, indicating that we're getting an increase in energy transfer most likely, and that is resulting in a change in ratio. And it's this change in ratio, like I said before, serves as a proxy for the GA and solution. And so we just can quickly jump to what happens when we put the protein in planta. And so we express this fusion protein in planta in Arabidopsis. And you're seeing dots here because I've targeted the sensor to the nucleus, so it's easier to discriminate the behaviors of neighboring cell types. And so that's why you're seeing these dots. And this is a light-grown hypocotyl. And the pseudovocal meristem is hiding in behind here. And already you're seeing sort of an interesting pattern. So it's like a Christmas tree, and we're not sure what this means. Are there really that many different GA levels, and why do some cells have higher or lower GA? So the first thing we wanted to do is make sure we're looking at GA patterning and not some other artifact. And so the way we do that is to create a non-responsive GPS1 that doesn't interact with GA, but has all the other components still intact. And therefore, if any changes that we observe in GPS1 but not GPS1NR are indicative of changes in GA. And so the way we can do this is to take the binding pocket of GID1, mutate it, and with residues that no longer will abolish GA binding. And we can also take mutate the GA interaction, the GID1 Guy interaction interface up here. So we can mutate Guy, and these are two glutamate residues that form salt bridges with positively charged residues in GID1. We can mutate these two to positively charged residues that will disallow that folding I was talking about earlier. Okay, so some results. GPS1 in vitro, if you, this is the starting ratio. If you add GA, you see an increase in this ratio, as expected. If we take some of the residues in the GA interaction, and GA interacting residues in the GA binding pocket of GID1, you can abolish this ratio change. So you get a sensor that no longer responds to GA in vitro. The similar mutation I made up here, described up here, mutating these negatively charged residues to positively charged residues greatly reduces the amount of ratio change you get. But neither of these two on their own is quite sufficient. So we want to make a completely orthogonal sensor that's a non-responsive. And so if we only mutate the GID, the DELA domain could be available to interact with endogenous GIDs. And if we only mutated the DELA, vice versa. So in order to get a completely NR sensor, we had to put both types of mutations in one protein. And that's GPS1NR. And this is a truly non-responsive in vitro and in vivo to GA changes. So what does GPS1NR look like when we express it in the light-grown hypocotyl? You can see that the patterning that we saw in the light-grown hypocotyl is absent in the GPS1NR, suggesting that these changes are indicative of changes in GA. And we can do another control to ask the question of what does it look like in a GA3 oxidase double mutant. This has the GA3 oxidase 1 and 2 double mutant has very low GA levels and all the expected low GA phenotypes. And this also does not show that distribution pattern that we saw with GPS1. We can quantify this by plotting some B-swarm and blocks plots of the nuclei in this region of the Arabidopsis hypocotyl. And you can see that there's higher ratios and an increased spread of ratios for the nuclei in GPS1 in Colombia. And then for the NR, we see lower ratios, and we lose that patterning, that spread. And the same is true for GA3 oxidase double mutant. We see a much lower ratio, and we lose that spread again, even though actually that is a GA responsive sensor. I'll stop here and say that we have to do, of course, a lot of biochemical characterizations of the protein. And we find that GPS1 prefers GA4 to the other GAs by at least an order of magnitude. So we're really, and GA4 is the dominant GA in Arabidopsis. So that's important for switching over to other plants, for example. It's also important for interpreting the results that I'll show you. And another important point for interpreting the results is that we find that GPS1 is not rapidly reversible with GA4 in vitro. It's a little hard to test this directly, but the indication is that it takes a while for the sensor to release the GA4 once it's bound. So that what we're seeing here is not necessarily, for example, in this cell that has a high ratio, that it's very high GA at that moment, but that either it has high GA right then or it recently had high GA. So we cannot interpret depletions of GA in the short term with this sensor. So just keep that in mind. Okay, so now we're back to the light-grown hypocotyl, and I can play a little movie. This is a 3D projection of a confocal experiment. And again, we see this with this spread of ratios, with the false coloration, we see high ratios, low ratios. And the first question we had is, well, we know GA is a growth-promoting hormone. Do these ratios correlate with growth, cell size? And in fact, when we look in just the region that we analyzed before, we do not see a overall correlation between cell size and GA levels, or the ratios, rather. And this is shown here. But then there's that other situation, that dark-grown seedling, where GA is very much required for that dramatic elongation of the hypocotyl. Is GA accumulation or GPS1 ratios correlated with growth there? And the answer here is yes. So as you take a dark-grown seedling and look at the spread of different cell section areas, so the larger cells do tend to have higher GPS1 emission ratios than smaller cells. And so actually, if you plot the entire dark-grown hypocotyl and ask what the ratios are, you can see that there's actually a very clear gradient of low ratios in the apical hook where cells are smaller, grading to higher ratios in the rapidly elongating region of the Arabidopsis hypocotyl in the dark. And so again, now we have these questions again. Well, now we see this may be an indication of where GA can be accumulating, but what does it mean? How is that gradient promoted, and what is the gradient doing? What is the relationship between GA and growth? So for the first question, what we're asking is, we'd like to ask the question of what's regulating GA. So just to back up a little bit and tell you that we actually do know quite a lot from a lot of work in the Arabidopsis community and other plants as well, of deciphering what's going on to promote this rapid elongation growth in the dark. So there's two key players, among others. The PIFs are these growth-promoting transcription factors that drive rapid elongation growth in the hypocotyl, and then GA is another one that's very important. And the way GA works is thought to be through just allowing PIFs to function by repressing the delta repressors of PIFs. So this is something we've known, and in the light, the situation is somewhat different. So you have phytochromes and other photoreceptors lowering the activities of PIFs, also potentially lowering GA levels, and both of these, the loss of both of these positive growth regulators results in less growth in the light-grown hypocotyl. But we saw that, we can see here that there's actually a couple open questions still. So what is generating the high GA levels in the dark is still sort of unknown. And then how do phytochromes actually repress GA levels in the light is also unknown. So we hypothesized that actually if PIF proteins are promoting GA levels, this would actually answer both questions, or potentially answer both questions. So accumulation of high PIF levels in the dark would promote GA, and then phytochrome repression of PIFs could lower GAs in the light. And so we simply just put the sensor in the PIF-Q mutant, which is a quadruple mutant in 4 of these PIFs, growth-promoting PIF factors, and we see that there is indeed less GA, as evidenced by lower GA emission and less GPS1 emission ratios in the PIF-Q mutant. And again, in the dark-grown hypocotyl, we don't see the patterning that we see in wild type or the PIF-Q in the NR mutant of the sensor, and also in the GA3 oxidase biosynthetic mutant. We can quantify this again with these B-swarm and box plots. And we can see that indeed that the PIF-Q mutant has lower GA levels or NLS GPS1 emission ratios in the dark-grown hypocotyl, whereas the controls are much lower. So there's two things here. Yes, there is a difference, but it's not a complete knockdown. So it's only part of the story. There's something else that is still keeping GA somewhat elevated in this dark-grown hypocotyl. But what we think we have now is that PIFs are somehow promoting either directly or indirectly GA accumulation in the dark-grown hypocotyl. And we think this represents a positive feedback loop. So we know that GA acts to promote PIF activity, and now we think if PIFs are promoting GA, it's a positive feedback loop. And maybe that's required for generating that really rapid elongation growth, this positive feedback loop through 2 positive growth regulators to generate this really rapid elongation growth, which is quite dramatic in the dark-grown hypocotyl. So just to summarize a little bit of what I told you already, how do hypocotyls elongate in the dark? we think we might have a positive feedback loop in hypocotyl growth. And interestingly, there's probably another positive feedback loop that's already been discussed by the Poppenberger lab between BR accumulation in the PIFs. BR and GA. So there could be multiple overlapping positive feedback loops here. But we're still asking the question of, we don't know how PIFs control GA distribution in hypocotyls, and we still don't know how do cells, other parts of the hypocotyl, know not to accumulate GA. So again, this is a mobile small molecule, so cells nearby each other have different ratios. We don't understand biochemically how this is occurring. I'm going to transition now to below the ground, because GA is, of course, also involved in root growth, including in Arabidopsis. So this GA1 biosynthetic mutant is dwarfed, and it has much shorter roots than wild type. So if you look in detail, GA affects root growth in at least 2 different ways. So this is a wild type root. The black bars are showing the start of the elongation zone, black arrows here. And you can see that GA1 mutant and GA3 oxidase mutant double mutant have smaller meristems. So it's affecting cell division or the maintenance of the meristem. And it's also affecting cell elongation, but not in a cut and dry way. So it's not that GA mutants don't elongate their cells, they do. And in fact, actually, they elongate from 0 here is the start of the elongation zone. They actually elongate faster than wild type for the first part of the elongation zone. And then they max out at a smaller overall cell length. And these aren't GA mutants. This is actually paclobutrazol-treated seedlings, so they have a GA biosynthetic inhibitor, but the effect is the same. And then in wild type, you see this nice even elongation, increase in cell length through the elongation zone, and then it maxes out as a higher overall cell length. So with that information in mind, where's the GA? What do we see with GPS1 in the Arabidopsis root? Where's the localization? Before I tell you that, I'll say that there's been some modeling work discussing of where the GA might be. And in this study, they hypothesized that GA might accumulate to high levels at the start of the elongation zone, and then as the cells proceed through the elongation zone, as they elongate, the dilution of the cytosol would actually lower GA levels, and that would be a signal to stop growth. So in other words, in showing this graphically, that from 0 in the start of the elongation zone, you'd actually see a falling of GA and an increasing DELA, and that would create this slowing of growth rate at the end of the elongation zone. And this is just simply actual measurements of cortical cell length through the elongation zone. So what do we see with GPS1? So this is right about here is the start of the elongation zone, where you can see the nuclei are starting to get farther apart. You can see that as we start the elongation zone, we see lower GA levels or lower NLS GPS1 emission ratios grading to higher ratios at the end of the elongation zone. So we actually see the opposite. We see that this patterning is not recapitulated in the 2 controls. And we can show this graphically and with the same plots, where we progress from the division zone to the early elongation zone to the late elongation zone. And we see an increase in variance and an increase in ratio of wild type with GPS1. And we don't see that same progression for the 2 controls through the elongation zone. And so the question becomes, what is the GA doing? And we still don't know the answer to this in terms of how GA is controlling or influencing specifically the spatial distribution of cell elongation. But the simplest explanation would be that GA acts locally in a dose-dependent manner. So as GA increases throughout the elongation zone, cortical cell length is also increasing. So maybe there's a sort of a linear relationship or a highly correlated relationship between GA and growth. And we now want to investigate to see if that model is actually correct. But to transition to the other question that we're interested in is what's generating the gradient in the first place, we have to go back to our old model of these are the different things that could be generating this gradient. So biosynthesis, transport, catabolism, for example. But we do have a little hint that when you take a GA1 mutant in Arabidopsis and you add GA, you actually can rescue growth. So exogenous GA in a GA1 mutant can actually potentially go to the right places to stimulate growth in the right ways. So what we can do is take Arabidopsis roots and add GA and see where it goes. And this treatment with exogenous GA would circumvent, of course, de novo biosynthesis within the plant or endogenous biosynthesis. So I'll play the movie one more time. And So you can take a look. These are roots growing in the root chip, which is a microfluidic device where we can treat with a compound and remove it. And at the beginning of this movie, the seedlings were treated with a dose of GA, and the GA was removed. And so I'll just freeze that at 20 minutes so you can get a better view of what happened. And we can see that the elongation zone cells did accumulate the exogenous GA, and the division zone cells didn't do so as much. We can plot this longitudinally. So this is from the bottom of the root. You can see in the division zone, we see not a lot of changes. The gray is before the treatment, and the blue is after. And as we progress to the elongation zone, we actually see a nice gradient of change that correlates very nicely with the original gradient, suggesting that import, or the ensemble behavior of import and catabolism, could be affecting a gradient that's very similar to what we see beforehand, suggesting that maybe that's what's going on. But we still don't know which of these or other potential biochemical components could be generating the gradient. So is it high import of the elongation zone that's generating this distribution gradient we're seeing? Is it high catabolism in the division zone? And of course, there's other possible factors that could be at play as well. So we're really going forward now to ask the question of what is actually happening in situ, in vivo, to generate this really interesting gradient. And of course, we can always go back again and ask the question of, if we find enzymes and transporters that are doing the job, what's controlling those enzymes and transporters to be where they are in sort of an asymmetric and distributed pattern? So in summary, I'll just say that GA gradients correlate with cell length in rapidly elongating root and shoot tissues of Arabidopsis. In hypocotyls, the gradient is regulated by light signaling components. In particular, I've shown you today the PIFs. In roots, this gradient is mirrored by the GA accumulation gradient, suggesting that import and/or depletion could control GA distribution in addition to de novo biosynthesis. And then in the future, we'd like to ask the questions of how does a complex growth behavior, such as photomorphogenesis, so that switch from dark growth to light growth, arise from dynamic hormone distributions. So we'd like to be able to image that sort of process and ask those questions. Right now, I'll stop and thank the people in my group that have contributed to this. And of course, some of the work was done in the Frommer lab when I was a postdoc, but Annalisa Riza and Ankit Walia are the two postdocs on this project right now in the group. And because I'm giving this talk because of the Tansley Insight review that I wrote, that was focused on ABA, and I'll just highlight that we do have ABA sensors as well. And we can treat roots growing in the root chip with ABA and see nice pulses of response, and then as we take the ABA away, the signal falls. And so we're really also looking at this dynamic here, and that's just a shameless lead into that are hiring for that project. And so if you're looking for a job or know someone who is, I'd really love to hear from you in the near future.